Usually when we talk about a medical diagnosis, there is this comforting expectation of precision. It feels almost like engineering, right?
Absolutely.
Like if you fall and break your arm, the x-ray shows that jagged white line on the black background, and the doctor just points at it and says, there it is. That's the problem. It's incredibly straightforward.
It's totally binary. The bone is broken or it isn't. The problem is visible. It can be categorized, and there is a clear mechanical solution to fix it.
Right. But then you step into the world of cancer recurrence, and suddenly that precision just completely vanishes. That X-ray machine becomes practically useless.
Yeah, it really does.
We are looking at a diagnostic landscape that is honestly terrifyingly murky. A patient goes through brutal treatments, surgery, chemotherapy, radiation, and the scans eventually come back completely clear.
Are they had to ring the bell?
Exactly. They get to ring the bell. They are declared cancer-free. It's the ultimate victory. But then there's this ticking time bomb. Months, sometimes a decade later, the disease aggressively returns.
It is the absolute definition of diagnostic muddy waters. The central mystery of oncology for decades has been, well, where does it hide for all those years? And even more importantly, how does it survive the constant around-the-clock patrols of our body's immune system?
Right, because your immune cells don't just take vacations.
No, they are constantly sweeping your tissues for foreign invaders.
And that mystery is exactly the mission of our deep dive today. We are unpacking a truly groundbreaking paper from Nature Cancer that reveals a shocking trick used by lung adenocarcinoma cells.
It's a fascinating study.
It really is. We are going to see that these cancer cells aren't just, chemically hiding from the immune system like we always assumed. They are literally physically shape-shifting their bodies to become invisible to the touch.
Yeah, and to set the stage for what we're looking at, we are exploring a phenomenon known as metastatic dormancy. This is that terrifying silent period where cancer cells have somehow escaped the original tumor in the lung, traveled through the bloodstream to a completely new organ, often the brain or the other lung, and just gone to sleep.
And the researchers managed to isolate the specific cells that do this, right?
Yes. In this study, they used experimental models, specifically human lung adenocarcinoma cells called H2087-LCC, and a mouse model called M802T4-LCC. They managed to track these primitive early escape cells that are uniquely capable of pulling off this disappearing act.
I am just utterly fascinated by this premise. The idea that cancer evasion isn't just about throwing up an invisible chemical cloak, but about completely altering their physical structural architecture. It feels, like science fiction.
It fundamentally changes how we view the physical reality of the disease. But to really grasp how they manage to hide in a brand new organ, we first have to understand how they break out of their original home. We have to look at the mechanics of the escape.
Okay, I love this. Let's look at the break-in, or the breakout, I guess, because tumors aren't just loose soups of cells, right? They are anchored.
Exactly. Normal epithelial cells, like the ones that line the inside of your lungs, are highly organized. They have what biologists call apico-basal polarity.
Which means what, exactly?
That just means they have a distinct top and a distinct bottom, much like bricks in a wall. And more importantly, they are glued tightly to all the neighboring cells around them by strong cell adhesion contacts. The most famous of these is a protein called E-cadherin. It acts like, heavy-duty molecular Velcro.
So to escape the lung, the cancer cell has to somehow unvelcro itself from the wall.
Yes. And it does this by running a specific biological algorithm known as EMT. That stands for epithelial to mesenchymal transition.
EMT, right.
When a cancer cell triggers EMT, it stops producing that E-cadherin Velcro. It loses its rigid top and bottom orientation, and it starts building these incredibly tense, rigid internal structures called actin stress fibers.
Okay, so if I'm visualizing this for you listening, think of it like a rigid, heavy brick wall. That's your normal lung tissue. But when this EMP program kicks in, individual bricks start transforming into flexible, spindly snakes.
That's a great way to picture it.
And these snake-like cells need a way to move, so they build internal bungee cords, those actin stress fibers, to physically pull, stretch, and propel themselves forward. They use that immense internal tension to literally squeeze through the microscopic cracks of your blood vessel walls.
It's a highly aggressive, forceful movement. In the study, researchers actually watched this happen in living tissue. They tagged these specific escaping cells with a fluorescent green marker, like GFP, and illuminated the blood vessels in mice with a marker called CD31.
So they could just watch them move.
Yeah. By day 7, they could see these green, spindly cells squeezing out of the bloodstream and invading the brain and lungs. Once they arrived, they took up residence by wrapping themselves around the outside of the blood vessels. We call this the perivascular niche.
So they ride the bloodstream like a highway, punch their way out, and then just hug the outside of the blood vessels in the brain. Wait, if I'm thinking about this logically, these cells are full of those stiff actin fibers, practically tense like loaded springs.
Right.
They are rigid, foreign objects just sitting on a blood vessel. Shouldn't your patrolling immune cells immediately recognize them as a threat?
That is the brilliant tension of this entire biological narrative. The very physical stiffness that gave them the strength to escape the primary tumor, survive the sheer force of the bloodstream, and push into a new organ.
That exact same stiffness betrays them.
Exactly. It makes them a massive, glowing target for the immune system. Being a rigid, tense object in a soft, foreign tissue is a guaranteed death sentence.
Okay, so let's do a quick recap and bullet summary of this first section for you listening. First, cancer cells escape by turning from stationary bricks into tense, migrating snakes via that EMT program.
Right, number 1.
Second, they use rigid actin stress fibers to move. And third, they settle near blood vessels in distant organs.
Spot on.
So here is a question for you to chew on. Think about your own immune cells patrolling your tissues right now. How would they react to finding a stiff, foreign object anchored to a blood vessel?
They'd want to destroy it immediately, which forces the cancer cells into a corner. To survive, they have to run an entirely new algorithm. They have to radically change their physical shape.
You have to drop the disguise and put on a new one. And this is where the master regulator comes in, right? A molecule called TGFβ.
Transforming growth factor beta. When these spindly, tense cancer cells settle into that perivascular niche, the local environment of the new organ naturally bathes them in this molecule, TGF-beta.
And anyone who has spent time looking at cancer research knows that TGF-beta is usually the ultimate villain. It's a huge paradox here.
It is a massive paradox. In the vast majority of aggressive late-stage cancers, TGF-beta acts like absolute rocket fuel. It usually makes cancer cells highly fibrogenic, meaning it makes them even stiffer, more scarred, and aggressively invasive.
Like, it just makes the bad guys multiply faster.
Yeah, exactly. If you look at aggressive late-stage cells, like the H2087SO line or A549 or 393T3 cells, they express a marker called SOX9. In those cells, TGF-beta just maintains this stiff, persistent, aggressive EMT state. But the cells we are talking about today aren't late-stage aggressive cells yet. They are those primitive early escape cells capable of dormancy.
Right. Our dormancy-competent progenitors express different markers like SOX2 and NKX21. And in these specific primitive cells, prolonged exposure to TGF-beta, specifically after about 3 to 7 days of being bathed in it, does something totally counterintuitive.
Doesn't hit the gas?
No, it slams on the brakes.
It forces them to go to sleep. But how does a chemical actually stop a cell from dividing?
It rewires the cell's internal engine. The TGF-beta signal causes the cell to drastically ramp up the production of specific inhibitors that act like brake pads on the cell cycle. Things like CDK inhibitors, specifically CDKN1A and CDKN2B, and WNT inhibitors like DKK1.
So it's just flooding its own engine with stop signals.
Exactly. And at the same time, the genetic markers for active rapid cell division completely drop off the map. A famous proliferation marker called Ki67 just plummets. Meanwhile, proteins associated with deep cellular sleep like P27Q1 shoot way up. The cell completely stops multiplying.
Oh, wow. And the researchers had to prove that this wasn't just a coincidence, right? That the environment was actually forcing this sleep state. To do that, they used one of my favorite methods in biology.
The knockout experiment.
Right. If you want to prove how a machine works, you break a piece of it and see what happens.
Yeah, they used a TGF-beta M-cherry reporter to first prove these solitary sleeping cells had active TGF-beta signaling. Then they used CRISPR technology to genetically delete the main receptor antenna for TGF-beta on the surface of the cancer cells, specifically a receptor called TGFBR2.
Okay, so what happened when they deleted it?
Well, the cells could still escape the lung and travel to the brain or the lungs, but once they got there, they were entirely deaf to the TGF-beta signal. They couldn't hear the command to go to sleep.
And because they couldn't go to sleep, they couldn't enter that protective dormant state, and they completely died out during the dormancy phase.
Exactly. Interestingly, they also tried deleting a different receptor, TGFbr3, and that only slightly delayed metastasis. It proved that TGFbr2 is the specific, non-negotiable key to entering the survival state.
Okay, so stopping division is only half the battle. The really mind-blowing part is the physical shapeshift. We have a great mnemonic to help you remember this transition. Think of the letters SSS. The cells go from being spindly to spheroidal to survive.
SSS, I like that. They lose that stretched out, tense snake shape and collapse into tiny, soft, round spheres. But genetically, they are in a bizarre halfway state.
What do you mean by halfway state?
Well, they don't revert back to being normal epithelial bricks. They keep their EMT escape genes turned on. It is an atypical EMT state. They're genetically primed for metastasis, but physically round and completely asleep.
Okay, let's do our second sub-theme recap and bullet summary here. First, primitive cancer cells react to TGF-beta differently than late-stage ones. Second, instead of growing, they hit the brakes and stop dividing. And third, they lose their spindly shape and become round, or spheroidal, while keeping their EMT genes turned on.
Perfect summary.
So question for you listening. If you were a cancer cell, would you rather be an aggressive, fast-growing cell that draws attention, or a sleeping round cell that no one notices?
Obviously the sleeping one. You blend right in. You become practically invisible.
But that brings up a huge mechanical question for me. It's one thing to say a signal tells the cell to become round and soft. It's another thing to actually do it. If the cell is packed full of tense, rigid actin cables, those internal bungee cords we talked about, how does it physically dismantle its own skeleton without destroying itself?
That is the missing link that the researchers uncovered, and it is a masterpiece of cellular engineering. They discovered the exact molecular scissor responsible for this demolition. It is an actin depolymerizing protein called gelsolin.
Gelsolin. It literally sounds like something that turns a solid structure into gel or jelly.
That is actually a perfect way to visualize it. To find this out, the researchers used a technique called single-cell RNA sequencing, or scRNA-seq.
Okay, explain that a bit.
Imagine being able to look at the active blueprints of a factory in real time to see exactly which machines are being turned on and off. As the cancer cells were exposed to TGF-beta over that 7-day period, the blueprints for making gelsolin spiked massively.
And what else did they see?
At the exact same time, the cell cycle signature dropped, and the cells transitioned into this soft, atypical EMT state.
Let's go back to our snake analogy from earlier.
Yeah.
You have this cell that is elongated, filled with tight, rigid internal bungee cords that are keeping it tense. The TGF-beta signal comes in, acting like the boss of the factory, and gives an emergency order to produce gelsolin.
Right.
And the gelsolin proteins act like millions of tiny chemical scissors running around the inside of the cell, frantically snipping all those tight actin bungee cords.
And when you cut a tense bungee cord, it snaps and goes completely slack.
Suddenly, the cell loses all its internal tension. The structural integrity just collapses. It turns into a soft little water balloon, held together only by a very thin, flexible layer of cortical actin just under its surface.
That structural collapse is the physical manifestation of the dormancy. They retain their dangerous identity on a genetic level, but they completely dismantle the physical machinery that made them stiff.
All right, time for our third recap. Bullet 1. TGF-beta triggers the production of gelsolin. Bullet 2. Gelsolin acts as scissors, cutting at the actin stress fibers.
And bullet 3. The cell loses its stiff internal skeleton and becomes a soft sphere.
Exactly. So here is the question for you. Picture that tense bungee cord snapping and going slack. How might losing internal tension change how a cell interacts with the things trying to touch it?
Which brings us to the ultimate question. Why does becoming a soft water balloon actually save the cancer cell's life? This is where we arrive at the most profound discovery of the paper. It completely reshapes how we understand the immune system. We are talking about a concept called immune mechanosurveillance.
Mechanosurveillance, meaning the immune system isn't just sniffing out chemicals, it's feeling for mechanics. It's using touch.
Precisely. We often get taught that the immune system is just a chemical factory, that white blood cells just swim by a target, recognize a bad chemical signature, and spray poison at it.
Like a drive-by shooting.
Yeah, exactly. But it's not. It is deeply, intimately physical. To clear a tumor cell, your body uses specialized soldiers, cytotoxic T lymphocytes, or CTLs, and natural killer cells, and they engage in close quarters, hand-to-hand combat.
They have to make contact.
They absolutely must grab onto the target cell and form a tight, physical, leak-proof seal. In immunology, we call this an immunological synapse.
An immunological synapse, okay.
Once that physical seal is locked in, the immune cell injects lethal cell-destroying proteins. Things like perforin, which punches holes in the target, and granzymes, which enter those holes and trigger the cell to self-destruct.
But here is the critical detail, and this absolutely blew my mind when I read it. In order to form that tight seal and physically push those toxins into the target, the immune cell needs leverage. It has to physically push against the cancer cell.
Yes, it needs resistance.
And for that push to work, this is basic Newton's third law here, the target cell must push back. It requires the target to have stiffness.
Without resistance, the synapse fails.
It sounds exactly like trying to hold a syringe with a needle and trying to violently stab it into a water balloon that is just floating freely in a swimming pool. If you try to stab it, what happens?
You can't pierce the surface.
Exactly. The balloon just squishes, deforms, and harmlessly pushes away from the needle. But if you take that exact same needle and try to stab a tense, rigid watermelon, the needle goes right in instantly.
That's a perfect analogy.
By using those gelsolin scissors to turn into a soft water balloon, the cancer cell makes it physically impossible for the immune cell to lock on and deliver the killing blow.
That is the soft armor. It is a brilliant, active mechanism of evasion. And the researchers didn't just theorize this, they proved it with some of the most elegant biophysical experiments I've ever seen. They used a tool called atomic force microscopy, or AFM.
Atomic force microscopy, basically a microscopic poking machine.
Real much. It uses a microscopic cantilever. Imagine a tiny diving board with an unimaginably sharp needle on the end of it. They use this tiny diving board to physically poke the cancer cells and measure exactly how much the cells push back.
They literally measure the squishiness of the cells.
Yes, they measured the stiffness in units called kilopascals. And they confirmed that once the cells were exposed to TGFβ and produced gelsolin, their physical stiffness plummeted. They became demonstrably softer.
But the real test is seeing what happens when you take the scissors away.
Right, the ultimate test. They ran an experiment where they used an shRNA to knock down or silence the gene responsible for making gelsolin in these dormant cells.
Okay, so without gelsolin, even when the environment was screaming at them with TGFβ,
the cells couldn't dismantle their internal bungee cords. They stayed rigidly tense.
they stayed watermelons instead of water balloons.
Yes, and the results were devastating for the cancer cells. The researchers mixed these artificially stiff, dormant cancer cells in a dish with active natural killer cells and OT1 CTLs.
And what happened?
Because the cancer cells were rigid, the immune cells easily locked on. They formed the immunological synapses without any issue. The immune cells degranulated, meaning they exposed a marker called LAMP1 and unleashed their lethal payloads like IFN gamma and TNF alpha, and they absolutely slaughtered the cancer cells.
Wow. It is literally life or death based purely on physical tension. If the cancer cell stays stiff, the immune system grabs it and kills it. If it goes soft, it survives indefinitely, just sleeping there in the tissue until some future event wakes it up.
The loss of biomechanical stiffness is not a side effect of dormancy. It is the core active cloaking mechanism against immune mechanosurveillance.
Okay, final recap for this section. Bullet 1. Immune cells need to push against a stiff surface to inject their lethal toxins. Bullet 2. By becoming soft, dormant cancer cells, evade this mechanosurveillance.
And bullet 3, blocking gelsolin keeps cells stiff, allowing the immune system to wipe them out.
Exactly. So what stands out to you about, as you're listening, is it the fact that our immune system uses physics and touch, not just chemical sensing, to fight disease? Because for me, it's just mind-blowing.
It really changes everything about how we approach relapse. When you look at all of this together, you realize that for decades, dormancy was viewed as a passive state. We thought the cancer cell was just tired, hunkering down like a bear in winter, passively waiting for a better environment to start growing again.
But it's not passive at all.
Not at all. This research proves dormancy is a highly active, dynamic, and heavily armored state. They are actively utilizing their environment to orchestrate a structural collapse purely to hide from the immune system.
It's a calculated defense strategy.
Yes. And understanding that opens up entirely new doors for revolutionary adjuvant therapies. Think about standard chemotherapy. Chemo targets cells that are actively dividing. So trying to poison sleeping, dormant cells is incredibly difficult and often ineffective. But what if we change the physics instead of the chemistry?
What if we develop drugs that specifically inhibit gelsolin?
Exactly. If we can administer a drug that blocks those molecular scissors, We could artificially force these sleeping cells to stay stiff. We would be ripping off their physical invisibility cloaks.
And then our own natural immune system could feel them again, lock on, and destroy them before they ever have a chance to wake up years later and cause a relapse.
We could clear the dormant cells entirely.
We spend so much time thinking of biology purely as chemistry. We think about hormones, DNA sequences, enzymes, and chemical signals floating around in a void. But diving into this research proves that biology is also deeply, undeniably mechanical. It is about tension.
It's about stiffness.
Right. It's about physical pushing and pulling at a microscopic level.
The physical forces shaping our biology are just as critical as the chemical ones.
They really are. And it leaves me thinking, if microscopic cancer cells are actively manipulating their own physical tension to trick your immune system, what other mechanical forces in your body are dictating your health right now, entirely under the radar?
That is a fascinating question.
Think about the stiffness of your tissues as you age, the mechanical strain on your organs, or the fluid pressure in your blood. How much of our health is governed by microscopic physics that we are only just beginning to understand? The next time you see a stark black and white X-ray, remember what you can't see. The softness, the tension, the invisible physics of the tissue, that might be exactly where the real story is hiding. Thank you for joining us on this deep dive. Keep questioning the hidden mechanics of the world around you.